Alright guys, welcome back to Underground Science, and in this video we'll be talking about the pathophysiology of COVID-19 part 2, alright? So we're literally going to pick off where we left off from part 1. Alright, so let me go ahead and pull up that page that we left off from. Alright, so one sec. Alright, there you go. So now let's keep on talking about what happened. So we said that it comes through this ACE2 enzyme, and it makes its way somehow through endocytosis into the type 2 pneumocyte. Alright, so into the type 2 pneumocyte, and we labeled that here. Now what happens? Well, now the virus is excreted out of this vesicle, and it's actually brought into the actual type 2 pneumocyte. Alright, so now we're going to see that the virus has released its genetic material, its RNA, into the type 2 pneumocyte. Alright, so this is its RNA. It's single-stranded, and remember, it's positive sense single-stranded RNA. So it's positive sense, means that it's already an it's acting like a ready mRNA to be translated into proteins. Alright, so let's go and see what happens next. So let's draw our type 2 pneumocyte bigger. Right, let's draw a type 2 pneumocyte bigger right here, like so. And we already know that it came in through a receptor, so we don't have to draw that. And let's draw our single-stranded RNA, all right, our positive sense single-stranded RNA. Next, what happens is that, remember in our cells, guys, we have our protein factory that we remembered as called the ribosome, all right, so but inside the ribosome, we have a large subunit of a ribosome, and then we have a small subunit of a ribosome. All right, so we have a large and small subunit of a ribosome. And again, we're going to go over translation, transcription, and protein synthesis in another video, in another playlist. So for now, let's not worry about that, and let's just worry about what happens, what SARS-CoV-2 does in the lungs. All right. So again, this is positive sense RNA, positive sense single-stranded RNA. All right, I'm going to fix that real quick. All right, so what's going to happen is that this RNA molecule can go in here, can go in the ribosome, and because it's positive sense, guys, it can actually get translated straight into proteins, right? So in some cases, it's going to get translated into proteins that are needed to build new viruses, right? So proteins that are needed to build new SARS-CoV-2 viruses. Or what's going to happen is that we're going to get proteins that make up the RNA polymerase enzyme, all right? So there's an enzyme right here present, right? So again, we can either translate this RNA material for proteins that are needed to make up the virus, or we can translate this material to make proteins that are needed to make up the enzyme called RNA-dependent RNA polymerase. All right, so let's go and draw that out. So we have another enzyme here that's called RNA-dependent RNA polymerase. I don't know why I drew it like that, but let's just go and draw it like this, all right? So we have an enzyme here called RNA-dependent RNA polymerase. RNA-dependent RNA polymerase. All right, so RNA-dependent, I don't know why I wrote a D there, but RNA-dependent RNA polymerase. All right, and when we talk about protein synthesis, we'll see that RNA polymerase pretty much takes DNA and makes pre-mRNA from that. All right, so what RNA polymerase is going to do, RNA-dependent RNA polymerase is so again, this ribosome can either translate this RNA material that was in the virus, all right, initially that was in the virus right here, the ribosome can translate this material into proteins needed to make up new viruses, or it can translate this RNA material to make proteins that are needed to make up this RNA-dependent RNA polymerase enzyme that's inside this type 2 pneumocyte. And what this RNA-dependent RNA polymerase enzyme will do is that it's actually going to synthesize more of this positive sense RNA, single-stranded RNA strands, right? So it's going to actually synthesize more. So it's going to synthesize, oops, sorry about that. It's going to synthesize more positive sense single-stranded RNA material that was initially in the SARS-CoV-2 virus, right? Okay, so that's what it's going to do. And specifically, if you want to know that RNA-dependent RNA polymerase, first what it does is that it takes those positive sense single-stranded RNA strands and it converts into converts it into negative sense single-stranded RNA. All right, so negative sense single-stranded RNA. And then it takes those negative sense single-stranded RNA strands and converts it back into positive sense. All right, so the difference, well, let me write this down. So the difference between a negative sense single-stranded RNA and a positive sense single-stranded RNA is that, remember that we said a positive sense single-stranded RNA acts like a ready mRNA that can be straight translated into proteins. All right, but negative sense single-stranded RNA cannot be translated into proteins. So it can't be translated into proteins until it's converted into a positive sense single-stranded RNA molecule, all right? So until it's ready with its 5' cap and a 3' poly A tail, pretty much a negative sense single-stranded RNA molecule has to be ready like a positive sense single-stranded RNA molecule, like an mRNA molecule, right, in order to be converted to proteins. But if it's just a negative sense single-stranded RNA, it cannot be converted to proteins right away, unlike our positive sense single-stranded RNA. So that's our RNA-dependent RNA polymerase's way of making more of this positive sense single-stranded RNA, more viral genetic material. All right, and then so now once we have all these new positive single-stranded RNA molecules, right, because of our RNA-dependent RNA polymerase. Remember that from our organelles, right? Remember our organelles in our cells. One of the organelles are called the endoplasmic reticulum. All right, so let's go and draw this out. So let's again go ahead. Sorry about taking so much space, but I don't want it to be too. I mean, it's already pretty ugly, but let's go ahead and try to keep on making it spatial. All right, so let's not try to like clog everything up in one space. So we have our positive sense single-stranded RNA, and remember we have these structures, these organelles in our cells called the endoplasmic reticulum. All right, so let's say it kind of looks like this. All right, it probably is a horrible drawing of the endoplasmic reticulum, but in our endoplasmic reticulum we have two types. We have a smooth endoplasmic reticulum and a rough endoplasmic reticulum. The rough endoplasmic reticulum it's called rough endoplasmic reticulum because it has ribosomes on it, right? It has way more ribosomes on it than a smooth endoplasmic reticulum does. All right, so let's say this is our rough endoplasmic reticulum here. All right, so these ribosomes are going to take these positive sense single-stranded RNA molecules. So let me go and keep on writing that down so we don't forget. Positive sense single-stranded RNA molecules. The ribosomes on the rough ER is going to take these positive sense single-stranded RNA molecules and are going to make these, and then are going to undergo translation, all right? So these positive sense single-stranded RNA molecules are going to undergo translation. So let me go and write that down. It's going to undergo translation, and so let me go and say that. And it's going to turn into proteins. Now, what type of proteins? Proteins involved are proteins that make up the SARS-CoV-2 virus. All right, so translation, not translations, and it's going to turn into proteins that are going to make up the SARS-CoV-2 virus. All right, so 4 proteins for the SARS-CoV-2 virus. All right, so you can see it's slowly hacking our own cells just to make more of itself. All right, so these ribosomes again are going to act as the place of translation, and we're going to undergo translation of our positive sense single-stranded RNA molecules. Then we're going to make the proteins related to, or make the proteins that make up our SARS-CoV-2 virus. And again, this is in our rough endoplasmic reticulum. So let me go and label the ribosomes wherever the ribosomes are. This is our rough endoplasmic reticulum. All right. So it's almost there. Now we have all the components needed to bud off new viruses. We have our genetic material and we have our proteins needed to make our virus, our SARS-CoV-2 virus, either from a ribosome in the cytoplasm that translates the positive sense RNA either to proteins, or remember, it translates this positive sense single-stranded RNA to make proteins that make up this enzyme, this RNA polymerase enzyme, so that the RNA polymerase enzyme can make more copies of the positive sense single-stranded RNA. Or we have proteins that part of the SARS-CoV-2 virus made by the ribosomes attached to the rough ER of the cell. All right, so now we are ready to package these materials, to package our genetic materials and our proteins that make up the SARS-CoV-2 virus that we made more of through translation, right? Ready to package them and bud them off, all right, into the alveolus. Remember, this is inside the type 2 pneumocyte, not the alveolus, technically, all right? So the type 2 pneumocyte is inside the alveolus, like it lines the alveolar membrane, but all this is happening inside the type 2 pneumocyte itself, not in the open cavity of the alveolus, all right? So let's go ahead and label this again. So this is a type 2 pneumocyte, all right? So now let's see so remember I just said package, right? So we have to package all these together to make the viruses. So hopefully package rings a bell, that we have another organelle in our cells, right? In general, that are responsible for packaging proteins, right? And packaging materials in order to transport those materials around the cell. And that organelle is called the Golgi apparatus, right? Or the Golgi bodies of our cells. So let's go and draw that out on a separate page, all right? So let's go to a separate page, and so let's again draw a type 2 pneumocyte right here. All right, and we have our Golgi bodies right here. All right, our Golgi bodies right here, and it probably doesn't look like that in a textbook or something, but let's go and just say that just guesstimate based on our drawing that this is the Golgi apparatus, all right, or the Golgi body, all right, so slash apparatus. All right, so now the Golgi body is going to take all of these proteins that we made, right? All these proteins that we made from the RNA-dependent polymerase or the ribosomes, right? Mostly the ribosomes though, all right? On the rough ER, we're going to take all these proteins that are going to make up the components of the SARS-CoV-2 virus, so like the S spikes and the protective covering around the virus, like the nucleocapsid and things like that. It's going to take these proteins and the genetic material, so our positive sense single-stranded RNA, take all these together, and all these materials are going to go inside the Golgi apparatus. The Golgi apparatus will package them together, and they're going to bud off vesicles of out of the Golgi apparatus of full made viruses, all right? So this is what it's going to look like. We're going to have new viruses with our genetic material inside our positive sense single-stranded RNA material, and we're going to have the proteins that we initially sent into the Golgi apparatus, and we're going to have our S spikes on our SARS-CoV-2 virus, and we're going to have many of these, all right? And these viruses are going to be ready to go and infect other cells, all right? So let me go and draw like a little angry face on here. All right, so this is what happens. This is how we bud off new viruses, and then this virus is going to bud off via exocytosis, right, of the type 2 pneumocyte. This is actually going to cause damage to the type 2 pneumocyte, all right, and that damage is going to trigger so many immune responses that we'll look at in the next video. But what's going to happen is that we're going to have many viruses that bud off, and that's going to come here into the open space of the alveolus, all right? So this is going to be now the open space of the alveolus, all right? And we'll pick up from here in the next video. So hopefully I see you guys there.